Hello, my name is Katarina Mandić and I'm a PhD student at the Ruđer Bošković Institute, Zagreb, Croatia. The topic of my poster is Multi-omics analysis of head and neck squamous cell carcinoma reveals non-overlapping epigenomic regulation of gene expression. In this study, we analyzed samples from head and neck squamous cell carcinoma patients. This cancer is one of the most common malignancies with a poor 5-year survival rate. And the role of epigenetics in cancer progression is still not fully understood. We integrated 3 omics data sets, microRNA transcriptome DNA methylation array, which were previously published, and complemented with gene expression data or transcriptome in order to answer the question, how does epigenetics influence gene expression? We performed exploratory analysis based on multiple sample annotations. The two heat maps show sample distance for microRNA and DNA methylation data. These results indicate that tissue, oropharyngeal or oral, and HPV status, positive or negative, do not distinguish samples. However, we see good separation based on cancer and control samples. This is further confirmed by the novel transcriptome data by principal component analysis, where we visualize the top 2 components, and we can see a clear separation of control and cancer samples. Nextly, we performed differential expression and methylation analysis in cancer versus control groups. We wanted to detect which genes are upregulated and which are downregulated in cancer. For expression data, we used BSEC2 library, and for DNA methylation data, we used RNBs. After adjusting for false discovery rate, we took a p-value cutoff of 0.5. In this table, we show the number of over and underexpressed genes, microRNA, CpG sites, and DNA methylation CpG islands. Interestingly, we observed that there are 3 times more hypomethylated CpG sites than hypermethylated, which indicates a genome-wide hypomethylation happening in cancer cells, and this has been previously observed in cancer. However, it's interesting to see that this ratio is not reflected in CpG islands. To integrate the omics, we took differentially expressed microRNA and mRNA and differentially methylated CpG islands. To integrate microRNA data, we searched for experimentally validated microRNA and gene pairs. And in cases where there were multiple microRNA targeting one gene, we took the one that had the highest Spearman correlation from the R expression data. And for CpG islands, we took those that appear in promoters of differentially expressed genes. Then we took gene, microRNA, and CpG island triplets and their respective fold change values and the quotient, which are represented in these 3 columns. And we performed k-means clustering on this data. And we observed that most clusters have only one of the epigenetic regulators. For example, in this green cluster, we can see a strong influence of microRNA and not so much for DNA methylation, as opposed to this darker green cluster, which has more hypomethylated CpG islands. And the opposite is observed for underexpressed genes. Nextly, we were interested in whether these clusters are functionally related, so we performed gene set enrichment with several databases: GEO, biological processes, KEGG, Reactome, and Transfect. And indeed, we found significant enrichments of functional gene set within these clusters. For example, we observed a notable enrichment of genes regulated by the E2F transcription factor family within clusters that exhibit differential methylation in CpG islands, such as cluster 1, 4, 6, and 10. This finding suggests a potential functional relationship between the E2F family and observed methylation pattern, highlighting their potential regulation of genes associated with these clusters. And finally, I would like to thank my collaborators from the Laboratory of Molecular Virology and Bacteriology and my bioinformatics group from the Lab of Machine Learning and Knowledge Representation that helped me do this work. Please contact me if you have any questions and come see my poster at the conference.